Double fertilization. This is Jimmy, the pollen grain. When Jimmy grew up, he no longer wanted to stay with his father, Mr. Anther. One day, there came a bee. Jimmy attached himself to the bee so that he could be transported onto another flower through bee for cross-pollination. Jimmy landed on the stigma of that flower. With this begins the process of double fertilization. The fertilization in flowering plants is defined as the fusion of male gamete carried by pollination with a female gamete present in the ovule to form a diploid zygote. For the double fertilization event to occur, the male gametes must land on the stigma. Once landed, it begins to germinate the tube cell, forming a long structure down the style and into the ovary. The generative nucleus travels behind the tube nucleus. Once it nears the ovary, it divides by mitosis to produce two haploid sperm nuclei. The pollen tube reaches the micropyle. It bursts and releases the sperm cells into the cytoplasm of the synergids. One of the two sperm cells travel from synergids to egg cell to fertilize with the egg nucleus, and this produces a diploid zygote which will become the embryo. This fusion is called as syngamy or true fertilization. The other sperm nuclei moves up and fuses with both of the polar or central nuclei, forming a triploid cell. Technically, this triploid structure is called as primary endosperm nucleus. It develops into an endosperm and serves as the embryo's food supply during early development. As three haploid nuclei are involved in the fusion, this process is called as triple fusion. Syngamy and triple fusion, both the processes are taking place simultaneously. Hence, together they are called as double fertilization. Double fertilization is the characteristic feature of the angiosperms. In this way, Jimmy gave his whole life to complete the double fertilization and for further development like fruits, seeds, and endosperm formation.